Okay, it seems that I didn't share the whiteboard in the last video clip, so I'm hoping now that I've solved the problem and it comes up. So I'm just going to see about putting on the whiteboard. Okay, so hopefully you can see this now. In the center of the board, you can see the word mitosis. Now, if we look at the bubbles, these are the connections that we can make to that word. So, for example, cell division, body and somite, and that cell division takes place in the body, or somatic cells, which is the technical name for body cells. It's important for the body's growth, repair and maintenance of those cells. And genetically, we need to make sure that each new cell gets the 46 chromosomes that it needs to function, so that a skin cell genetically is identical to its neighboring skin cells, and that the liver or the kidney cells are genetically identical, so that new kidney and liver cells can be made. Now, if we look at mitosis, this only forms one small part of the cell cycle. Most of it is interphase, and if you look at the notes that I've used in the PowerPoints, you can see that the vast majority of the cell cycle is the interphase, where there is a growth, a synthesis stage, and a second growth stage. But within mitosis, there are 4 stages. Those are prophase, in which the nuclear membrane dissolves. So we're up here at the top left. And the chromosomes appear. Normally in a cell, you can't see the chromosomes in the nucleus because they have to condense, and they have to coil, and they have to compact, so that they can be moved when the cell divides. Okay, so the centrioles are essential because they produce spindle fibers. Now, these are made of protein, and the centrioles begin to move towards the poles. That's the opposite ends of the cell. And the spindle fibers at the same time attach the centriole to the chromosome. Now, in metaphase, the centrioles are now positioned at 2 ends of the pole, and you have these spindle fibers that have dragged the chromosomes to the center of the cell, which we call the equator, or more technical language would be the metaphase plate. Now, at anaphase, the centrioles now help to contract the spindle fibers, and because the chromosomes are in pairs, we call those chromatids, and the contraction of the centriole and the spindle fibers causes the 2 pair chromosomes to be pulled apart into single chromosomes. Now, these are identical, and they're pulled towards those poles. Now, in telophase, because we've now finished with the movement of chromosomes, they can uncoil, and as they uncoil, they dissolve and disappear, and a new nuclear membrane goes around our DNA and chromosomes to trap it in there once more. The spindle fibers are no longer needed because the chromosomes have now moved, and at the same time the cytoplasm begins to split, just as if you're getting 2 pieces of dough when making a large loaf and splitting them into 2, so the cell starts to divide. Now, the process is known as cytokinesis, and it's really the end of mitosis. And then we split the cell into two portions, and we have two daughter cells which are completely identical. And those cells go on into interphase, where they grow, they synthesize new organelles, they grow a little bit more. And when they're mature, they can go through the whole process again, all right, to make a new body cell. Okay, so essentially mitosis, 4 stages: splitting of the chromosomes so they get an equal share of the chromosomes, 46 chromosomes, genetically identical, all right, and it's for the body cells, and it allows for growth in our early stages of our development up until about 21, and then repair and maintenance of the body. Okay, now if I slide this over, and hopefully you can see this, okay, then we're looking at meiosis, or meiosis, if you want it spelt correctly. So the process, all right, is essentially, and it's essentially the same as mitosis. And the chromosomes move in a similar way to mitosis, except in the second division of meiosis, instead of being pulled this way, the chromosomes, they're actually pulled at 90 degrees. But it's a small kind of like speciality of meiosis 2. Now, at the end of meiosis 1, instead of going into interphase, those two daughter cells now go through a division again, which we call meiosis 2. So there's no doubling up of the chromosomes like at the beginning. They go straight into meiosis 2. And that means that the 46 chromosomes now get divided again. So in meiosis 1, we have prophase 1, metaphase 1, anaphase 1, telophase 1. Then they immediately go through into meiosis 2, and each of the daughter cells produced goes through prophase 2, metaphase 2, anaphase 2, and telophase 2. What that means is that the original cell, and this takes place inside the testes and the ovaries, now produces 4 daughter cells, but with half the number of chromosomes, which we call the haploid number. Now, if we go right to the very top, this arrow here, it says, what's the biological significance of this process? Well, remember, this is for sexual reproduction and to form the sex cells, the gametes, which in the male is the sperm, and in the female, the ovum, or the ova, if it's plural. Okay, but normally only one of the ova is produced. So you have one egg or one ovum each month. Now, although essentially the chromosome movements are the same, there is an important difference, and that's why I've run prophase 1 in red. And if you look in prophase 1, the chromosomes, they pair up. So they're like that. They're kind of like paired up, just as before, but this time they cross over like that. They become what we call entangled. And it's at that point that the genes are randomly swapped between the 2 sister chromosomes, or chromatids as we call them. All right. Now, that means that the swapping of genes lead to small variation in our species as humans, and anything that reproduces by sexual reproduction, whether it's a dog, a cat, a crocodile, an oak tree, whatever, that produces by sexual reproduction, these small variations are very important. Because when the male gamete unites with the female gamete, in our case the sperm and the ovum, The 23 chromosomes from each parent fuse together to form a fertilized egg. That contains 46 chromosomes, which then starts dividing by mitosis to produce a fetus and embryo. Now, that means that because of the random assortment in the male genetic sperm and the female genetic ovum, that means we get a total random assortment of our genes, and that's why we all end up differently, not looking like our mum or dad or our brothers and sisters, because it's a total random assortment that occurs every time. So we've got thousands upon thousands, millions upon millions of variations. And that's why in this world of ours, out of nearly 6 to 7 billion people, no two people are the same. Okay, difference in eye color, hair color, shape, okay, skin color, but they are just random variations. And this is what drives evolution. Okay, so not only is it just the formation of the gametes and the fact that you get 4 daughter cells with half the number of chromosomes, the haploid number, but it's the driving force to evolution. So hopefully that's now shared. Okay, I can drop that down. You see me back, and we are sorted, hopefully, for the revision for your in-class test. Okay. Sorry for the mishap last time. Hopefully it's now recorded and we're back on course.